And then Karandeep is followed by 2 or 3 flash talks here of some of the projects that are coming out of UCSD, so we get a better idea of what's going on. Okay, so over to you.
Do you want the subtitles up or no?
No.
How do you take them off?
Yeah.
Okay. Thanks, everyone. It's good to be back, and it was good to talk about all the big things we want to do with large language models at the bedside to make things better. And now I'm going to talk about experiences of trying to actually implement AI and all the actual challenges that come with it. The idea here is not to say that we are setting out to fail, but to share some lessons of failure so that we can think about what are some of the issues we might be able to anticipate and fix. And I'll close with how we can do better. Here are some of my disclosures. And I want to start with what I consider to be the AI paradox in health, which is that researched models aren't implemented and implemented models aren't researched. So if you talk to researchers and you look at the scientific literature, you'll come across a lot of studies that describe models, lesser studies that validate models, even lesser studies that talk about models that are actually implemented, and way fewer studies that talk about how well an implemented model was actually adopted. And so you might come away with the impression that there's just not much happening in the space of healthcare AI, because in the research side, it's all about model development. But if there's a set of pipeline that comes from the kind of research pipeline, there's this flood of stuff coming from industry. And so the reality is that a lot of the healthcare AI solutions that health systems are using across the country, because they're not academic medical centers, are not coming from researchers, but they're coming from companies. So what is an AI implementation? So AI implementation, when I say AI implementation, I'm actually talking mostly about AI models that produce a recommendation that get reviewed by a patient or reviewed by a person, usually a clinician, and used to drive a clinical workflow. This is not the only way that AI is implemented, but it's a really common way. This can relate to not just images, it can relate to just EHR data. It can relate to other kinds of data, like in pathology. And another common way that AI gets implemented is this, where you start off with an AI model that produces a recommendation, and that recommendation actually changes the workflow, and it's used to reorganize a person's work. So just an example of this one, at Michigan, when I was there, we have rapid response teams that proactively go and check in on patients, even when they're not getting alerted, to do so. And so they wondered, how can we spend our time wisely? What we do right now is we call out to each of the nursing units and ask the charge nurse, who should we check in on? And that takes a long time. It's pretty inefficient. So what we ended up doing was building not one, but 2 or 3 different scores that predict every patient's risk of going to the ICU. And then what they do is they actually go in and check in on the highest 10 to 15 patients at risk of going to the ICU who are currently not in the ICU. So that actually drives their workflow. It reorganizes their work. They spend their time differently. Doesn't save them any time, but the idea is if it's a useful model, it's a good model, they'll spend their time more effectively at the patient level and allocate their time better. So an ideal AI implementation is one that produces recommendations that are informative and can be acted upon, allocates time more effectively, and it allocates resources towards the people who actually stand to benefit from that. And that's where we think about things like equity. And in reality, it's not always this simple. Models don't always produce recommendations. Sometimes they produce risk scores, and then we kind of apply our reasoning skills on top of those risk scores to say we want to use that to drive interventions. But many times, models actually nowadays are shifting towards producing recommendations because they're relying on things like causal learning, where you can predict individualized treatment effects and who stands to benefit most if you intervene. So why do AI implementations fail? Largely, they fail for 2 different reasons. One reason is that the models themselves are problematic, and there are issues with either the models or the recommendations. The second thing relates to issues with workflow, how care gets reallocated and reorganized, and the issues with that, and the actual change management associated with trying to actually change real workflows in healthcare. Okay, so here's not an exhaustive list, but some of the things we'll touch on for why AI models fail. We'll start with an example, and then we'll dive straight into kind of lack of transparency and work through each of these examples. So a couple years ago, we were starting to pilot the Epic sepsis model, a proprietary sepsis model at Michigan in 3 nursing units. We were working with our sepsis team at Michigan and finding that as we were rolling this out in those 3 units, the nurses actually said, you know, you gave us some numbers about how good the model is based on what the company told you. It just doesn't seem as useful as what you told us. We were also hearing from the sepsis committee, we're tracking our performance measures and we're tracking our bundle compliance, and nothing has really budged in those 3 units since we started using this. So we looked at those 2 things together and we said, let's independently look at this. And we're not going to look at the patients where it was used to drive decisions because that creates circular logic and problems. Most of the hospital wasn't using it. So we said, let's look at the rest of the hospital and see how well this model actually performs. Okay, so basic facts. What goes into this model? The model is trained at 3 different institutions with data from 400,000 patients. If you look at what it used to predict sepsis, which is a kind of a body's response to a really severe bacterial infection, it uses things that largely make sense: vital signs, medication orders, lab values, comorbidities, information about demographics like age, all things that I think if you asked a clinician, they would say, yeah, those all seem relatively reasonable. And if you look at how well this model does, you don't have to understand what this number means, but it achieves an area under the curve of around 0.8, which is relatively good. There's a whole issue around, what is a good AUC, and there's not one value that's good. It depends on the use case. But just believe me that if you presented this to a chief medical information officer at most places, they would say, oh, above 0.8, that's good. We can use it. So what's the first transparency issue? The first transparency issue is, how did we even print this as a supplement to our paper? We had to actually ask Epic, because the actual information sheet says proprietary and confidential. So, we're getting ready to publish. The reviewers are like, can you share information about this algorithm? So we said, we can ask. And so we went and asked Epic, and they said, sure, you can share. And this isn't the only thing that was in there. It was, maybe 3 or 4 pages or something. But this was kind of a paragraph lifted straight from their information sheet, just describing some of the basic things that go in the model. But the final model is 80 variables. And you can see here, we only know kind of a handful of them. The 80 variables are not spelled out. So one aspect of transparency that's problematic in these algorithms is we often actually don't know what the ingredients are that go into them. This is a paper out of Nigam Shah's group at Stanford, where they looked at a lot of the Epic models, and they looked at the model cards, they're kind of information sheets that Epic gives you, and they said, how complete are those information sheets? Each row here is a different reporting guideline, and each column here is a different model. And I'll focus just on the sepsis column here, but you can look across the board. And I think what you'll appreciate is about half the information is there, regardless of kind of which tool we're talking about. So even if you have the information sheet from Epic or from any other kind of company, even some of the research models, there's often key information that you need to know to kind of drive how we're going to use this clinically. Does this make sense to use? That's missing from there. And so therefore, you're working with incomplete information. The other part of the transparency thing here is that, what about the patients? So if the model card is proprietary and confidential, I have never gotten a clear answer on, can I share that with patients? Can I present that information to patients? And I'll come back to that, because that's actually a real issue that I think is a problem. There's nothing in that model card that's like secret. I mean, it's like all obvious stuff that could easily be shared. And when I told this to Epic, they said, we know that this actually does get shared. I said, great, then maybe you can just make it available. And they said, no, that's just not how we do it. And that's not a critique of Epic. Actually, a lot of other companies are in the same boat. They have a secret sauce, and they don't want to release the secret sauce. And I get that, and I support that. But a lot of the stuff that was on there, there's no secret sauce there. That's basic things that, those are the ingredients. We can debate about the recipe. The recipe is the secret sauce, but come on, the ingredients are just the basic things we need to know about and be able to talk about. So how well did the model do? We ran it silently at Michigan for a couple of years. And we then went and said, how well does it predict the onset of sepsis, which is effectively what the model was supposed to be doing. According to Epic, this is predicting sepsis before it happens. And when we looked at it, the area under the curve was 0.63, which is actually substantially worse than 0.80. So it's 0.5 is the worst you can be, basically. 1 is the best you can be. So a drop from 0.8 to 0.6 is like meaningfully worse. Interestingly, if you take the scores in the 3 hours after sepsis has occurred, it's actually almost exactly what Epic reports. So we said that's an interesting discrepancy. Maybe the sepsis becomes more obvious. There's something weird happening here. But we said, let's file this away. We couldn't explain this because based on the model card, there's nothing in the model card that really explains why there would be this discrepancy, other than just us thinking the sepsis becomes more obvious in those 3 hours, and that's why the model has a better chance of picking it out. But it was not just, so is this a reproducibility issue? So you could say, well, the Epic evaluation was at different hospitals, and this is at Michigan. So we had actually asked Epic, can you run your tool at Michigan and tell us how well this model does at Michigan? And they said, we have great news for you. The model does amazing at Michigan. It's even better than what we published in our white paper. It's an area under the curve of 0.88. And I said, that's interesting because we're running it and getting 0.63. What outcome are you using? And so, at that time, some of the consensus criteria hadn't been updated, and so they were using some kind of made-up outcome. We weren't using the perfect outcome. We were using a composite outcome, but that's largely what's responsible for this difference, as was the issue of timing, because it was like, when is someone septic? What is the time zero? And in sepsis literature, people debate all about that, but it's really important. Like when you're using algorithms in the hospital and you're trying to predict future events, you have to make sure they actually are future events, and they're not things that are already in motion, even if the timestamp hasn't happened yet. Okay, so this had a transparency issue, this had a reproducibility issue. Is there a transportability issue? So in AI, there's transportability is the idea that if an algorithm works in one setting, it might not work in another. So we have to transport the algorithm there and see if it works. And so you don't just take it as a given that you take a system that works and it works elsewhere, particularly if you're trying to estimate risk of something, where there's a lot of practice patterns baked into the algorithm predictors, baked into the outcome, and so you can't anticipate. So we went to, I called up a friend at WashU, Patrick Lyons, who's now on the West Coast at OHSU, and I said, hey, can we look at this at the Barnes Jewish Hospital Network? Because he was in a position where he could look at that data. He worked with Philip Payne, who leads one of their informatics centers at WashU, and we were able to look at 9 of their hospitals where they were running this score silently and see how well does this tool do. At the upper end of it, it's somewhat close to what Epic reports. If you just look on that y-axis, that's the area of the curve, we're close to 0.75. On the lower end of it, 0.55. And I think one of those is actually the Barnes Jewish Hospital. So the lesson here, and actually if you look carefully in the middle, there's that orange dot marked Hospital J. It's not in the paper because they wanted to take it out, but I put that here because that's Michigan. And I want to highlight that, this was not some cockamamie outlier at Michigan. This was the average performance. And in fact, there's a pretty big range around it. And this was actually using the latest kind of consensus definition of sepsis. Okay, so there's a reproducibility issue here. There's a transparency issue here. There is a transportability issue. So let's go back to the transparency issue. A lot of these issues that I said are actually reproducibility issues or transportability issues. They're actually transparency issues because it turned out an investigative journalist called around and found out that one of the medication orders that was in the model was a prescription of antibiotics. That was not known to us. And had that been known to me, we probably would have never wrote that paper. We would have just said, aha, that's why it's happening. And we wouldn't have even gone through the work of looking at it because that would have, or it would have changed how we looked at it because we would have said, okay, it's useful for these tasks, but not for those tasks. And the task that it was being used for was to decide if you should start someone on antibiotics. So here's what happens. Person comes in, they have a fever, they're tachycardic, they don't look good, clammy. Clinician says this person has sepsis, starts antibiotics, that person's epic sepsis model score goes up. The sepsis model detects sepsis or predicts sepsis and tells you, hey, you should start antibiotics. And so essentially that circular logic was responsible not for the entire issue, but for large swaths of the issues here. Now there's been places where this model has been used where it's worked. So I don't claim that this is true across the board, but just to say that these kinds of transparency issues often hold us back because if we don't know the ingredients, we can't really even think about the recipe. Another quick example of this is DeepMind had published this paper in Nature, where they said, we have a state-of-the-art model to predict acute kidney injury. And what they specifically said was, future work should now prospectively evaluate and independently validate this model and look at how it improves clinical utility. So, I looked up, I said, this is great. I was super excited when this paper came out. I had a long Twitter thread about it. And I said, great, let me jump to the code availability statement and data availability statement. The data availability statement is, we use data from 700,000 veterans. Go talk to the VA if you want the data. Interestingly, they got the data actually off of the VA servers onto their cloud, which is not going to ever happen again. But they were able to get that for this study. The other piece of this was their code availability statement, where they said, we invented TensorFlow, which is a popular deep learning library, or it was popular. And then they say, here's the link to TensorFlow. So really, there's no way to recreate this stuff. So to actually recreate this, you would have to get this data from 110 VA hospitals across the country, write your code to be able to clean the data in the same exact way they did it, use the VA resources to actually do this because you can't get the VA to give you their data outside the VA firewall, and then spend all this time trying to replicate this model. So that's what we did. So I actually had a contract at the time with the VA to build an acute kidney injury model when this paper came out. So this is one of these situations where one part of the VA is not talking to the other part of the VA, and so we were like, well, why are you paying us to do this when you just had Google do this? But that's fine, we'll still do it. So we dropped everything we were doing and we said, let's try to replicate what Google did. We actually released the model online, it's on GitHub. We released a software package that lets you clean data in the way that they did, and that's also on GitHub. And the reproducibility issue here was we weren't able to include billing codes in the note or in our model, and so we couldn't quite get the level of performance they got. But the more interesting thing was, depending on which VA hospital you're at some hospitals, the model is useless. At some, it actually looks quite useful. But the individual experience of a clinician at a VA is not going to depend on the composite VA. It's going to depend on where they work. So transportability issue and reproducibility issue. Okay, so those are all model things. I know we're deep in the math and stuff, but another basic thing that's a math thing that I won't get into is, can a model actually be worse than no model? And mathematically, the answer is yes. Area under the curve isn't a good way to figure that out. There's actually other ways to figure that out. There's a person at Memorial Sloan Kettering who came up with this concept of net benefit, where if you quantify a trade-off that you're willing to accept between false positives and true positives, so people we would treat unnecessarily, inappropriately versus those who deserve treatment, that your model can actually act in a way, perform in a way that's worse than doing nothing. And so there's ways of doing that. And so that's something that we're trying to get people to look at before they start using models when they're still evaluating them. And then the last model thing before I jump into some implementation things is predicting non-modifiable risk. A lot of things in healthcare are very, very difficult to change. So an example is this. If you're trying to prevent people from getting readmitted to the hospital when they've gotten discharged, what should you predict with an AI algorithm? Should you predict the risk that they're going to get readmitted? Or should you predict the chance that a readmission could be avoided if you intervene? 99% of the research on this has been on the first item. And the issue is every health system independently finds that when you roll out a readmission prediction tool and you target your interventions at the highest risk people, the highest risk people still come back because most of their risk is actually non-modifiable, has to do with the underlying illness they have, their chronic disease, comorbidities. And so that doesn't work. Being able to do the second, there are methods now that allow us to do that, especially if we have the right data and we've previously tried different things, we can actually learn from those different things. And so a lot of effort now, not just for readmission, but for all kinds of things, is going into this idea here of, you're coming up at an intersection. Don't tell me the probability of stuff. Should I turn left or should I turn right? That's what I want to know. And maybe I want to know, is it 2 minutes longer if I turn left than if I go right? And maybe I still want to go left, even if it's longer, because there's less traffic lights and it's easier to drive. So that's the kind of thing that's starting to come into health, but still relatively early when it comes to the kind of prediction of actions we should take, which is a little bit different than things that Eric is talking about with multimodal prediction, that kind of thing. This is about what should we do clinically? Give me the right advice to be able to guide someone. Okay, so let's say you've got the best model. Why could it still fail when you try to intervene based on it and try to change the workflow? So the first one is just because you've got a good model doesn't mean your intervention works. People often assume that when they have a good model that I'm deploying my AI. We don't deploy AI, we deploy workflow changes. The AI is something that helps us allocate how we make those changes. If we're talking about diagnosis, it might help us figure out what the diagnosis is. If we're talking about sepsis or some kind of in-hospital outcome or outpatient outcome, it might help us guide what we should do next or what we should order or how we should counsel that person. But that counseling, if the counseling is not effective anyway, then guiding the counseling or deciding who to counsel based on AI ain't gonna work either. So I think there's a lot of disconnect there sometimes where people kind of go from we've got a great tool to not thinking about how you actually bring that tool and link it to something effective. You need an effective intervention for this to work. Workload's a big issue. This is actually the percentage of patients that generated an alert using a sepsis model at 24 hospitals across the country, indexed to day 0 of COVID. So if you look beforehand, about 8% of patients were generating sepsis alerts based on kind of following the same set of rules across 24 hospitals, 4 health systems. The day COVID hit, the number of alerts, actually the percentage of people generating alerts went way up. Why? Because everyone had a fever, everyone had a high heart rate, and they had COVID, but for all intents and purposes, they were indistinguishable from bacterial sepsis to a sepsis algorithm. Not only did the percentage of total alerts go up, which I'm showing here, the actual overall number of alerts went up. And that's remarkable because we had fewer patients hospitalized. We cleared out and canceled elective surgeries. So people were looking at the parking lot and saying, these hospitals are empty. We had fewer patients, but they were a heck of a lot sicker and generating more sepsis alerts. Just giving you a sense for, what it felt like. And actually at Michigan, 3 weeks into our first case of COVID, the nurses said, can we shut off these sepsis alerts? And we said, sure. And they said, this is kind of like there's that cartoon where there's this dog and there's like a fire all around him and he's like, it's fine. And that's what the nurses were, right? They're like, everyone has COVID. I don't need an alert for anything. I'm good. Like I know what's going on. Readiness for change is a really, really big one. When it comes to things like, let's say fall screening. Well, if you're a magnet hospital, you are judged. One of your things you're judged on is the number of falls that your patients have. And so most hospitals have pretty intensive fall screening protocols. They screen everyone for falls at different frequencies. And so we were looking at this, not, camera-based, but just EHR-based fall screening tool that just looks at numbers in your EHR and says, you're at risk to fall, you're okay. And we said, and then actually nurses came to us and said, we want to use this model. And I said, okay, so let's say we use this model. Who are you not going to screen now? because the model says they're low risk. And they said, oh, no, we're still going to screen everyone. So then I was like, what's the model going to do then? If you're going to screen everyone, there's no place to reallocate care. And at the end of it, a lot of the AI and the way it's used in clinical workflows is, again, to reallocate or to change the workflow and the way care gets delivered. Another one is resource constraints. And I teamed up with Nigam Shah at Stanford and Andrew Vickers at Memorial Sloan Kettering, and we did this kind of thought exercise, which was people commonly report out like, oh, my model has 90% sensitivity, or they like all the different measures. And we said, that's great. If you have a model that predicts who needs to go to the ICU, and you only have 2 open ICU beds, what if your model says 6 people need to go? Are you still 90% sensitive? Like, if the tree falls in the forest, what do we believe? What the model predicts or what we can actually do? And the reality is that at constrained hospitals, of which if you look in our, ER at La Jolla, we are not immune to this. If there is only so much room, no amount of AI can help. Because if AI, you're trying to use it to give people more care, but you have limits on what you can deliver, your realized performance is actually much worse than your predicted performance based on whatever math you did beforehand. So that math goes out the window. And so you can try to figure out, if you're trying to do an intervention, go talk to the people who are going to deliver that intervention and say, how much work can you actually take on? And if they say we're already bursting at the seams, then all you're thinking is, I'm going to reorganize your work. If they have capacity to take on more work, then maybe you can do some of this kind of stuff. But we're not going to say no to someone when a clinician says this person needs to go to the ICU. That's often very difficult to say no to that. And because we can't say no to that, we're never going to go to a system where we can fully rely on things like AI to guide us here. So what can we do better? How can we make AI better and safer? So some of the things, this is my experience at Michigan where I led AI governance for the last 5 years, was we have to have a way to track what actually is being used. It's really important for a lot of stakeholders. It's important for researchers, important for innovators, important for healthcare leadership, important for students. And so we had kind of a model card set of dashboards that was basically, here's everything that's being used, who it's being used by, and what state of activity it's in right now. And we would actively evaluate things. So we were very active in governance. And I think that actually paid off because as we looked at multiple different sepsis models, multiple different deterioration models, we would publish stuff when we find it doesn't work. We'd reach out to companies and say, this doesn't work as well as you think. And in the case of Epic, they actually completely overhauled their sepsis model in the last year. Now, granted, they said, we started overhauling it like a month before our paper came out, but it happened just very recently. So it's been a multi-year project, and maybe it affected their priority level to some degree. But in any case, this is an entirely new model. Their actual information sheet now is 8 times as long as it was before. So a lot more informative. They actually took antibiotics out of the model. They now have an explanation for why they took them out. But the idea was, now it makes a lot of sense. And now it's a much more trustworthy model as a result. The FDA plays somewhat of a role, but as kind of Eric was alluding to, FDA is really not that active in this space as you might think, based on their kind of FDA clearances and all the stuff that they release. So the Epic sepsis model, as of today, still not FDA cleared, still not FDA regulated, even though this headline right here says FDA says AI tools to warn of sepsis should be regulated, and that's the FDA document that says that. So why is it not there? Because it's an EHR, it's regulated differently than companies selling models, and so they've decided we're just not going to worry about the FDA. Obviously, the executive order happened recently with Biden, and so there's a lot of interest now, and there's a lot of kind of calls for public comment in this space. And even before the executive order, this is a public comment kind of period that was coordinated by the DHHS, Department of Health and Human Services, around issues around algorithmic transparency. And so one thing I would urge folks is, whether you're an innovator, whether you're a health system person, this is stuff that will affect you. And so, this is stuff that we should try to respond to, especially if we've got something to add. And the message along with, I think, 60 or 80 other colleagues put together for this was, we just need to make sure that those proprietary information sheets can be made available to the public. I don't see any reason why basic ingredients of a model, not the recipe, but the basic ingredients can't be made available. And I think that would go a long way towards, we want trustworthy AI. How can it be trustworthy if I can't tell you whether it's looking at the color of your skin to make a prediction? And that sounds like it's funny, but UCSF published a study on how the Epic no-show model at one point was using religion as a predictor. So it's not unheard of. This kind of stuff happens. And the only way I discovered it was I read the sheet and I was like, holy crap. And I Googled it and I came across Bob Wachter and Sarah Murray's work, and 60 health systems were still using that model at that time. So it's not like they suddenly shut it off. 60 systems around the country were still relying on it. Things we can do, and I'll close soon. One is we can have better reporting guidelines around AI. So a lot of the old guidelines are coming from the statistic literature that don't really touch on a lot of the issues around more recent models like transformers. And so these are two kind of examples of reporting guidelines that I was involved in their development. Tripod AI is accepted and will be published soon. Probast AI is being submitted soon. But I think this is kind of a call for just higher quality reporting of algorithm performance. And the last thing I'll close with is just participating in partnerships. Like, we're not alone. Partnerships I've historically, participated in include the Health AI Partnership. And we're going to be taking an active role in the Coalition for Health AI, where they're trying to set up assurance labs where we can actually try to benchmark these tools. We have no idea how a model does other than the PowerPoint deck it's printed on. So I think this will give us some better sense of how well things do so that we can actually use them effectively. Thank you so much.
A very, very thoughtful presentation. Lots of new ideas, insights. I think it really points to the way things are going. So I love this CHAI, chai, right? Like, especially afternoon. And your title is also the Chief Health AI Officer. This is also chai. CHAI. So thanks for bringing some chai to UCSD. So I think we have like 2 or 3 presentations, flash presentations. Unless somebody has a pressing question to ask Kardeep here. Yes, there's a question.
Thank you so much for the presentation. So, you mentioned that it looks like it's hard to use AI in the clinical settings. And it looks like what you looked at is using it for sepsis, using it to predict, readmissions. And these are like great things to look at, but these are set by like CMS. Like, these are metrics set by CMS, and they're old. So just like Dr. Krupal said, I mean, we usually screen people for like once they get to age 50. We look for, I don't know, PSA. So should we look for other stuff in the hospital to use AI for, and not focus on these metrics that are, I'm not sure they're good, but it looks like they're just set by CMS for certain reasons, more for like hospital grading, reimbursement, etc. Does that make sense?
Yeah, it makes sense. So here's what I'll say to that. Absolutely, we should look at using it for other things. This morning, that's why I talked about some of our generative AI pilots we're launching, because I actually think chart review and information extraction from charts for all kinds of things is fertile ground for the use of large language models. And that's why we're launching, I think, 15 pilots right now for large language models for all kinds of things, not just, hospital-based things and metric things, but, a lot of other even qualitative things where people are just spending a lot of their time and energy that we can avoid. Here's what I would say, La Jolla is bursting at the seams. And so there's only so many things you can do. It is true that a lot of readmission stuff has failed before. We have a lot more tools now available to us. But if you look at how can you fix that problem, you can smooth out transfers out of La Jolla to another place and figure out who actually is safe to transfer and make those transfers happen faster. When a bed opens up in the hospital, because someone got discharged, you can try to fill that bed as fast as you can and figure out like the turnover stuff. And bed assignment today is not automated, it's fully manual. There's phone conversations that happen. And even at like Cleveland Clinic, the average time to fill a bed is like something along the order of like 3 hours once the bed opens up. So there are these micro opportunities to improve. And just like the example that we had earlier, where the hospital's really busy in December, but like light in July, and so people kind of make all their plans in July, and then they deal with the consequences in December. We have the same thing happen every day. Like the hospital's horrible at 7 a.m., people get discharged, ER gets emptied, and then like we're okay, and then it kind of gradually builds up again. So I think, unfortunately, the only two things we can do are we can get more beds to fill, and that's why a lot of decisions have been made around trying to actually get more beds to be able to fill. And a lot of the other things are reshuffling. So I wouldn't look at those as CMS metrics. I would say preventing a readmission is freeing up La Jolla. So is smoothing out a transfer. So is filling a bed that's empty. All these things will add up. But these are the things that historically are not research fundable. Historically, these are the things that health systems don't invest in. So we're trying to see, can we take the best of what's out there and improve like the day-to-day functioning of the hospital, both with stuff we're building in-house and with kind of partners in industry.
I had a quick question on the readmission and sepsis model. You know how you had mentioned, well, those that have high morbidity and mortality or just high-risk patients have the highly likelihood of coming back to the hospital. Are there models that are present for like too early for discharge AI models where, hey, you're gonna send this guy home, he's gonna come back in 2 weeks, or when does that BPA actually fire for the risk of readmission, and then what action does physicians take on for that?
Yeah, so I think BPA, which stands for alert that you essentially get when a model has high risk, is probably the wrong way for us to intervene on a lot of this stuff. There are algorithms that predict your estimated day of discharge, and that drives a lot of what case management does. And there are algorithms that predict risk of readmission at the time of discharge. So there are models for everything: risk of going to the ICU, risk of coming out of the ICU. I think where the opportunity is the people who built those algorithms wrote the paper and then they left. The opportunity here is to actually see, can we try to actually use that to change our workflow and see if it actually helps. And that requires very tight coordination between folks who understand what's happening at the algorithm level and folks who are in healthcare leadership. And I think that's something that we have at UC San Diego that actually not a lot of places have.
Thanks, Karthik. Thanks. I think you gave a lot of ideas, especially for PhD students among here to launch research papers. Okay, so now we're going to follow up with 3 talks that are what we call flash talks. These are startups, new ventures, or even pre-venture kind of stage. The first of them actually follows very nicely with Karthik's talk, the warning of the Epic system, but how to do it right. So that's the effort from UCSD, led by grad student Aaron Lucina and PI Shamim Namadi. And Aaron is going to take us through that. That will be followed by a couple of others, Professor Sanjay Sinha and Professor Ed, are going to talk about some of the work they are doing over there.
I wanted to thank you all for having me. It's actually a huge honor to follow such titans in the field. I saw a lot of friendly faces in the audience, a lot of people that allowed Kilcijo to really come to life. So I just wanted to share a bit about that story, a bit about the research that's led to the formation of the company, how UCSD supported us in that journey, and how we hope to give back in the horizon. So I'm Aaron. I'm a PhD student in the bioinformatics department. I was fortunate enough to join this team led by Professor Shamim Namadi, Dr. Atul Malhotra, as well as Dr. Supreet Shashi-Kumar in the Department of Medicine. Before joining this team, I worked at Genentech and Illumina for a number of years, and then got into data science for clinical trial consulting. And I was supporting an early phase 2 drug for congenital adrenal hyperplasia for the endocrinologists in the room. And I was supporting a drug safety monitoring board. And I became really interested in this process because it was so manual and so slow. We were generating reports that would then go to a panel of experts, and then months later you might say, hey, this patient had an adverse event. So I became really interested, like how can modern AI, modern deep learning techniques actually support this process of identifying adverse events? And so I called Shamim Namadi. I basically Googled that, like adverse event prediction in hospitals, and he was the top result. And I called him. I called him out of the blue. And then after just one conversation, I was so enamored, I actually quit my job on the spot and then decided to go for PhD, which is what I'm doing now. So there was a great question in the last talk about, why even focus on sepsis? And a lot of this talk's gonna be about sepsis, not exclusively so, but some of it. And I want to differentiate a little bit to address that point. There's things that are hospital-related measures like the SEP1 bundle that it's CMS mandates and says you have to report this. There's readmission mandates, but under some of those, sepsis remains the #1 cause of mortality and morbidity worldwide. And for every hour you can save in identification and administration of antibiotics, you can actually reduce mortality. Time and time again, studies show reduce mortality by nearly 8% every hour. And so that's the fundamental thesis here. Can we actually train AI to identify sepsis ahead of time? And then, you don't deploy AI, you deploy workflow solutions. Can we deploy a workflow solution that will allow for early intervention and hopefully improve patient outcomes? So this is the work that took place at UC San Diego before I joined. And it's this paper in Nature's Digital Medicine called Artificial Intelligence Sepsis Prediction Algorithm Learns to Say, I don't know. The basic summary of this paper is that it's a modern, novel deep learning technique using a feed-forward neural network and some extra stuff to basically flag out of distribution samples. And the takeaway message is the performance was very good. So this became kind of the state of the art for sepsis prediction. And there's a head-to-head comparison in this paper if you'd like to look with the epic sepsis model that Dr. Singh just talked about. But I think, and as Dr. Topol was mentioning, there's all these models that sort of exist and are almost collecting dust on a bookshelf. They're on everybody's laptops. They say, hey, we have like really good performance, but ultimately very, very few of those actually make it into the clinic. And so that's really what my work is centered on, and what eventually led to the formation of Symposium is how do we actually build an ecosystem and a platform that actually allows you to take those models that are developed and actually put them into clinical practice. So right now, for every single patient in either the La Jolla or the Hillcrest area, if you were to walk in the door, we essentially are able to get their data in real time. And we're able to actually pipe that to a platform that's able to integrate a lot of multimodal sources, including patient notes, including wearables, and actually put models on top of that then close the loop in the EHR. So hopefully none of you guys have to go to the emergency department later, but if you did, we'd actually be able to type that in and actually use predictive models on your particular data. And I think this is interesting because there's a lot of challenges. We're talking about the unimodal models. We're talking about difficulties of actually deploying, but and I see some experts in wearable technology in the room right now. This is something where you can basically have a wearable and you can basically pipe that in and it would actually be able to flow directly and match with EHR data, and then you can build models on top of that. We talked a lot about large language models. This is actually something we're doing right now for patients that come into the emergency department. We actually have a large language model doing differential diagnosis on the notes. And that's not happening through things like OpenAI or ChatGPT. This is all happening in HIPAA-compliant enclaves using locally trained large language models. So I want to talk about how that idea, that concept of the platform, bring it back to sepsis and show this fundamental thesis of early detection in a real patient. So this is we had kind of a deployment plan of we're going to operate silently across 60 care units. That's wards, ICUs, emergency departments at UC San Diego. And we're going to look and see, is this model working as we expect it to? A lot of times you train these models retrospectively, there's data drift, there's things like, is it actually meeting the workflow, as in like, it's actually predicting before administration of antibiotics, those sort of details. And so what I have here, it's a little bit confusing, but the top this is the real patient timeline. And the bottom is sort of our counterfactual, what would have been if our sepsis model went live? And so what you see here is they're in the wards for about 4 days. They have some vitals that become aberrant, so maybe their heart rate elevates to like 89, they're not tachycardic, but it's trending in the wrong direction. Then the AI essentially jumps from a score 7 to 81, doesn't matter, it starts going really high above the threshold. And what really happened in this patient was nothing. So for 5 hours, for 6 hours later, labs get ordered, the vitals are critical, and then sepsis gets confirmed 7 hours later. So our idea here is, okay, if we had been live, if we had made a workflow intervention at this point where the model passed the threshold, done that BPA, that's the pop-up, and said, somebody should take a look at this patient, somebody should potentially get involved, then you could have ordered the lab results, and you potentially would have saved about 5 hours. And if you remember that 8% for every hour saved, that's about 40% increase in survivability. Okay, so that's all well and good. This is a silent prospective implementation, but there's at this point in time, at least prior, there had never been a deep learning model for sepsis that had actually gone to the clinic and actually shown improvement in patient outcomes. And so this is really our big contribution of this work, was that we actually did that. So we went live December 2022. We've been live for over a year now. And our paper got released a couple weeks ago, where we actually showed that after deployment of the model, we had about a 17% relative reduction in sepsis-related mortality. So this is adjusting for all confounders, patient severity, et cetera, and it seemed to have a significant impact in actually early administration of antibiotics, improvement in step 1 bundle, and most importantly, patient mortality. And what's neat about this is we did kind of like a mechanistic look at this. We looked at cases where the best practice advisory fired, powered by the AI, and how did the nurse respond to that? And when we looked at those cases where they said, oh, this might be a problem, and they escalated to MD, there was a significant reduction, substantial reduction in time to antibiotics. So basically, in cases where the BPA fired and people took it seriously, care was given sooner, and these are true positive cases, so this was basically improvement in outcome. So that's a lot about the research, and I want to talk about how that work that happened at UC San Diego really fed into the story of the company. So we licensed this technology from Tech Transfer Office. Chris Lorryman was in the room earlier. We owe him a lot for just being very supportive on that. And we formed Skillsesio, and then we went through a kind of a student-run incubator that started in Boston, but had its first year a couple years ago in San Diego. That's called the Nucleate Program. That's the logo on the right here. And we ended up actually getting the grand prize for that competition. And so after that, we've since gone on to get multiple federal investments, government grants. We won another quick pitch through something called New Fund, and our kind of served as an inflection point for the growth of the company side. And so I really want to share maybe the biggest news on the company side, which is that we applied for a grant, phase 1, phase 2 fast track STTR for this work, and we actually received it. So that ended up being about $1.7 million for the company. That's allowing us to take this research that kind of started, nucleated at San Diego, and bring it to other institutions. So we're in active talks right now with 3 other academic hospital systems to deploy the sepsis AI at their institutions. I also think it's a good example of how this forms kind of a virtuous cycle. UCSD is the subcontractor now on this Skillsesio grant. So it came out of the university, and we're actually able to feed it back in through federal investment Skillsesio can apply for, and they give it directly to the university. And I want to talk about, we talked about this horizontal expansion going across multiple sites as a big goal. We're also really moving into a vertical expansion. So obviously sepsis is just one of many potential application areas. And so here's kind of like a roadmap that we have. We have multiple models running in silent mode at UC San Diego, including things like deterioration, need for mechanical ventilation, and respiratory failure. And we have a study that I'm really excited about where we're actually giving Fitbits to patients and syncing it up with their EHR data. And the idea is to then help with predicting readmission. So I'll keep it brief because it's a flash talk. Just want to thank you for your time, and I'll take any questions.
Thank you, Aaron. As always, a terrific presentation. It's a tough act to follow. Any quick question for Aaron?
Hi. It sounds really exciting. So this works with Epic or with the electronic medical record. So how did that integration work, and how would future applications, how complicated would it be to work with them?
Thank you so much for that question. Can I go back?
Yeah.
Go back somehow.
Yeah, got it. Okay, well, I can't pull it out for you, but yeah, thanks for that question. Yeah, it's not good. One of the big things you're really cognizant of is there are solutions that exist that require clinicians to leave their workflow. So you would go to some other website and basically take you away from care of the patient, your common routine in hyperspace. We're really cognizant that this is a closed loop solution. So actually the way it integrates is same way that an external lab would integrate, or same way that a bedside monitor would. We write a message, which is like a device message, back to the EHR into a flow sheet landing spot. So what's nice about that is just as you might have like a heart rate recorded in the flow sheet, you would also have AI sepsis risk score. And so what's nice is you can imagine anybody building any model on this platform, and it would actually, you could just write to a new flow sheet row. So that's essentially how the integration works. And then when you want to build workflow logic on top of that, then you can, the institutions Epic analysts can basically build a patient list or best practice advisory or whatever suits the need. Any other questions?
This is really cool stuff. I was wondering in terms of, I actually had a very similar question. In terms of integration and like working together with people, what was it that the clinics that you were working with that made it possible for you to integrate where a lot of other, groups struggle to actually integrate these types of things?
Yeah, I mean, I think the answer is, I'll say twofold. One, it's this systems design is maybe just a core competency of our group and something we're very cognizant of. So it's a cloud-based system. We leverage serverless computing. We're very familiar with healthcare interoperability, HL7v2, FHIR, all the standards. So that's just a knowledge base that helps in that. And then the other piece that's more important maybe is just the institutional support. UC San Diego is just incredibly, UC San Diego Health, Chris Longhurst, everyone from the health IT team, at the time Andrew Breeze, just incredibly supportive to basically push the project forward.
That's awesome. Thank you so much.
Thank you.
Thank you, Aaron. Yeah, thank you. Our next talk is going to be a slight shift. It's not as further along. It's actually an early stage idea, but I'm really pleased to present faculty member Professor Sanjay Sinha, who's actually at Sanford Burnham Prebys. Just joined them from the East Coast, from the NIH National Cancer Institute. So really pleased to welcome him here. Also because we're trying to go broader than UCSD, bring the whole La Jolla Mesa here, Saul, Sanford, and everyone. And hopefully this also gives you an encouragement that some of you are thinking about ideas, continue to develop them. Maybe next year you'll be giving this talk here. So let's invite Sanju here. Yeah, thank you, Sanju.
Thank you. I would like to start by thanking everyone for their time and listening. The lab that I started here actually focuses on developing new drugs that could actually act as a preventive therapy for cancer. However, I would talk about today a technology that I developed in the last year or so time while my time at NIH and NCI, and actually take it to a different spectrum of tools where we would use data, but use really tools that are even far advanced than LLMs. It's called linear regression. Okay. Really, really simple tools. So the goal of this tool that I developed in the last year or so time is to identify what is the in-depth mechanism of action for small molecule inhibitors. If you have worked with drugs, this is almost a ubiquitous problem that the drug that you are utilizing in various phases of clinical trials, even as far advanced as phase 3, you still find multiple off-targets as well as toxicity. And the key reason is that currently the industry as well as academia is really not well equipped with the set of tools and full knowledge about the small molecules and what does it precisely really targets in your cell. So we developed this new technology that's called Deep Target. And for a given drug that you have, it can do the following things. And then I will tell you how it does that. So it aims to predict what is the primary target of your drug that is it's driving its mechanism of action in the disease that you are starting with. Or, for example, in this particular case, we are focused on oncology and primarily the cancer type that you're starting with. And I will go furthermore in details. Second, it identifies which mutation can actually target for a given target or a gene that you are starting with. And finally, it identifies an ability that we are very excited about is the secondary targets of the drug that is mediating its efficacy in different diseases. For example, and I will give you a few examples of anti-malarial drugs that actually have efficacy in anti-cancer efficacy, and we are showing how does it really show this efficacy. Not only one disease to another, from a certain subtype of cancer to a different type of cancer as well, is it driving by a different, completely different target as well. The key idea behind this is actually quite simple. The idea is that, let's say you have a small molecule that is targeting, is this the one? Can you guys see this?
Yeah.
It's targeting a particular protein and inhibiting it. The idea is that instead of actually inhibiting and giving by the drug, you can actually directly delete the target gene which is making this protein. and doing that via genetic methods like CRISPR. Deleting this target gene can actually mimic the drug treatment effect, and thus, if you can do this and create a similar data for drug as well as via CRISPR, you can actually mimic the drug treatment effect. So let's say that you have some like 500 or so cell lines, right? Some of them are resistant to the drug, some of them are actually sensitive to the drug. And in those 500 or so cell lines, you knocked out every single gene, right? The gene that is supposed to be the true target of the drug would actually mimic the drug response that you saw during the treatment. If this principle is true, when you make this comparison, a very simple comparison, you can actually identify what is the primary target of the drug. And not only that, what is the probability that each gene might be the target of this drug. But this principle was not made by me. This was almost made like a decade ago. And what we did is really utilize this principle and used it to take it to the next step by not only identifying the primary target, but using the natural variation that is already occurring in the cancer cells, where some of these cells have a mutation because these are cancer cell lines, and some of them don't have a mutation. So let's say that you have a drug that only targets a particular mutation, right? Then you will only see this matching that I was just referring to in the cell lines that actually have the mutation. And thus, you can actually really take advantage of such a conditioning regression, almost to identify drugs that specifically only targets the mutant-specific mutation form of a particular gene. And this is very interesting because actually in oncology, it is quite desired to really target a precise mutation. If we use this principle, we actually identify almost all the mutant inhibitors that currently exist out there in clinics. But not only that, we have far new number of drugs as well that can precisely target new mutations as well as existing mutations. And I will show you a few examples. And finally, you can again target the existing natural variants in the cancer cell lines to identify what if the primary target that the drug is not targeting, that the drug is targeting, what if that gene is not at all present, right? And the drug is still showing efficacy. How the hell is it really showing efficacy in those cell lines? And thus, we actually only analyze those cell lines to identify what is the secondary target of that drug. What is the secondary target of that drug that is really mediating its efficacy? Now, while Eric was referring to this, that anyone can make predictions, the most important contribution that we really made in this whole effort, in my opinion, is actually we spent almost an year to find what is the real gold standard drug target pairs. And this is a very major problem. And if you want to take away one thing from this whole thing that you can do far better than us, is to take these gold standard data sets with you, okay? And test your own models on these gold standard data sets. And we made it publicly available rather than actually Google saying that our data is available on request or never making it available, like Karan was referring to. Because we believe that this will advance furthermore, and these models will go far beyond what we are developing at this stage. Finally, what we observed is that really using these very simple principles can actually help us identify information about the drug that when we integrate it into clinical data, we found that it can really give us a clinical potential that I will show you some data on. So what is a single slide results that we have right now? The first one is that we found actually there are 101 drugs out there, where 21 drugs that are currently given to patients in clinics, either in trials or FDA approved, that our model predicts have an incorrect target. The doctors are given to are currently giving, but we think that we are giving it to X, but instead it is targeting Y. We are pointing out here one such drug, ibrutinib. This is FDA approved first-line therapy for multiple blood cancers as well as stem cell transplantation. It's identified that it actually targets this gene called BTK, and it does target a gene called BTK in blood. However, we found that actually in all the solid tumors, it actually targets this completely different gene that causes lung cancer actually, and can also cure lung cancer and has huge potential for that. Just showing you data here that when your cell line has no mutation versus has this mutation, this EGFR, it can actually really kill those cell lines, specifically lung cancer cell lines. Beyond, we actually picked up 2 anti-malarial drugs that's been found in the last 6 to 7 years in Dana-Farber, actually, that these are anti-malarial and can be given to pediatric cancer children And they can be given at such a high dosage, they're almost as if that their sweat could be orange in color. And this drug is actually orange and given in like ice cream, and children love it. It actually makes the taste of ice cream better. Really, that's what one of the output in the clinics that have been claimed that the children asking for more such ice cream. There is anecdotal example actually that someone tried to commit suicide using overdose of this drug, and guess what? The patient survived, because actually it's so hard to actually overdose on this drug. That's how much you can give it to the patients. Okay, we found that its anti-cancer efficacy is primarily driven, completely unknown, by our tool, by actually targeting mitochondrial complex 1 and 3. That's completely novel mechanism of action that we have identified, and even now working towards not only advancing these drugs, but identifying new drugs that could be targeting these pathways. And final piece of information that I want to share with you, that I think in some way really epitomizes this idea of using really simple principles on big data creatively, in my opinion, that could really reflect back that how much these data could teach us, is that when we use this single similarity score that I just referred to in the previous slide, and computed it for all the small molecules that have been tested or are out there publicly for EGFR, this gene that is right now one of the major genes or major oncogenes in lung cancer and a few other cancer types. And we computed this for 50 different small molecules that can actually target EGFR. And here on x-axis, I'm plotting which phase of the clinical trial they really reached the small molecule retrospectively in 20 years. Okay, and the y-axis is our score. And you can look at this plot and see that how the single score can actually tell us far distinguish the drugs that have eventually reached and passed all phases of clinical trials and got approval from FDA, versus the drugs that never even reached any clinical phases. Imagine if we would have had this particular data 20 years ago. The number of patients that we could have really saved, and the number of amount, not only financial burden, which is far aside, the amount of burden that we would have really been able to save from the patients would have been, in my opinion, really, really amazing. But secondly, that comes out of this data is that this particular single plot that shows that in phase 2, some drugs really failed. However, these drugs are the ones that we think may have failed due to various different reasons. And potentially we can actually pick them up, go back and find why did they fail, right? And our score really shows that they have a high clinical promise, and maybe go back and revive them potentially. We think has a high potential. Secondly, it not just applies to this particular gene called EGFR, it also extends to multiple different targeted cancer therapy genes. MAP2 kinase, MET are a few genes that I'm just noting here. We think this score is very imperfect. I just want to make this note. We do not claim that this score really tries to solve which drug will go further in the clinical trials. However, we have a very modest correlation of 0.4 almost. So on an average, I would say that we have a very modest prediction right now at this stage. However, still in some cases, we succeed quite well, and those are the cases we want to focus on. And rather go back and learn from these clinical trials, what could we do better? How could we do further? This analysis also revealed something very interesting that, trust me, I would have never expected when I actually started this. This drug called osimertinib. This is a first-line therapy that is given to lung cancer patients that have this mutation called T790 mutation. Okay? We found out that this drug actually do not actually bind or even target that mutation. This is currently given in clinics, not just in trial. It actually got approved from FDA. We actually found out that it only targets and eventually binds to L858R mutation, that it also is approved for, by the way, okay, but not to T790 mutation. We do have another different candidate that we are right now progressing, but the fact that there is a first-line therapy that targets something else really shows that how ubiquitous this problem is. And I want to tell you how did we really find out. This is not actually a prediction that came out of Deep Target, okay? Because if Deep Target would have given us this prediction, I wouldn't have given it to my collaborator. I have to be honest, okay? This is so common that is given right now too. However, this was actually our positive control in our experiment. We thought this must work, okay? And then let's see how our predictions will do. And our positive controls did not work, okay? That's how ubiquitous this problem is of drug target misidentification. The final thing that I want to say is that out of our model, there was a lot of intriguing data that came out of for multiple different drugs. and we have no damn clue how to really do with that or how to interpret it very carefully. So we are actually looking for people who are experts of different domains who want to work with us that can help us really like decode or deconvolve how does this data to interpret it and do better in that. With that, I will close and thank you for your time.
That was a great talk. Do we have any questions or comments for Sam? Stephanie?
Yeah, I'm just curious of. Thank you, very interesting, promising platform. So I'm wondering, what is the source data to predict or actually reveal the mechanism of action?
Sure. There are a couple of different data modules that go into the model. The first one is actually drug response data across cancer cell lines. This is publicly available data for DepMap that is being used. This data that was released from Broad Institute. The second is genetic screens that have been created in the last few years, and a few other omics data from the same cancer cell lines. So these are deep screening data and omics data that has been created on cancer cell lines of these drugs and of this deep omics data that has been generated that we integrate, try to identify this. This is the major information that is passed in the model. There is a few deep learning component as well that I did not talk about. We use the structure information and a few other, but what I really wanted to talk about is this simple algorithm and this data is the most important factor of identifying the drug target. That's what we have been found, at least we have found.
Thank you, Sam. Thank you. So our last flash
Just
talk.
one last quick question.
Sure, please.
So just this week, there was an article published around thalidomide, which was taken off the market as an antibiotic for pregnancy because it causes birth defects. So the famous led to restructuring the FDA. This week it was published that it's a chiral molecule, so there's an L and a D isomer. And one of them is effective and the other causes all the birth defects.
Oh my God.
So my question is, do you know if there's any chirality in any of the CRISPR-Cas series?
Actually, that's a very good question. From the CRISPR-Cas, we don't know if the chirality will play the role, but there are, of course, the Cas5s that can have chirality. But very interestingly, I'm aware that actually, if you take the same drug, these drugs come out as almost like powder, right? So in these screens, if you take them from different vendors, it makes a huge difference because the production quality of it makes a crazy difference. So chirality might be playing a role there, that how you're really producing these powders and how it plays a role eventually. By the way, I just want to tell you, I told you only the success story, okay? There's a couple of cases that we predicted that were absolute garbage, okay? Just want to be completely transparent about it. And we do think that production quality of these things and how that makes a difference. But chirality is a very important point. I have not taken that into account. Thank you.
It would be very interesting.
Thank you.
Thank you, Sanjay. And we'll go to the next talk. It's not pulling up here.
If it works.
What's that? Yeah. Professor Ed Wang, who is assistant professor in ECE and the design lab here upstairs. So he's going to be talking about his new company, Brilliant Labs, right?
Yep.
So maybe, let's see, it'll come up. Why don't you get introduced?
I'm the literal opposite of the far from home. You guys are in my building, hanging out with me. Thank you all. Kind of nice. My lab is on the 3rd floor and the company's on the 4th floor, and I'm on the 2nd floor hanging out. So this is a new experience for me. It's kind of fun.
Why are you not going up?
I don't know. I think I made it an hour ago. Maybe that's the problem.
Okay, you want to just introduce the concept?
Yeah. Well, I guess we don't need slides. So my name is Edward Wang. I'm an assistant professor with a joint appointment with the School of Engineering, the ECE department, Electrical Computer Engineering, and also with the design lab. And so design lab is a really human-centered design approach. And obviously I'm an engineering professor. And so I combine the two sides. And our approach at my lab, which is called Digital Health Technologies Lab, really focuses on thinking about the end application, which is health monitoring, but really designing new engineering systems that can approach that. And in my lab, we work on sort of a variety of different things. The thing that we'll really focus on today is around smartphone-based health monitoring. But we also work on wearable devices, both on the sort of data side, but also designing the device itself. And obviously, how can I be a digital health lab if I'm not working on LLMs? But today we're going to talk about smartphones. I guess my slides will never work, so we'll keep talking. Should I wait? Should I just keep going? Oh, this looks promising.
Yeah. Let's see.
I'll wait for a hot
So
second.
far, everything was going smoothly, and then suddenly,
Yeah. Otherwise, you guys will have to use your imagination, which is okay. I just came from teaching, so hopefully I still got it. All right, that's fine. So yeah, so what's really nice about, I think, being a lab here at UC San Diego, and I think for me particularly really cool, is that my lab being on the 3rd floor, we invented this new way of monitoring blood pressure, and hopefully the slide will come up. But recently we were able to form a company called Billion Labs. The real vision is to leverage the smartphones in everyone's pocket. There's billions of phones in the world, and we can transform each of them into a health monitor. And since then, since the forming of this company, we've been able to license the technology from UCSD and then continue developing that, and even acquiring an NIH grant recently to be able to actually take this through sort of the first stages of clinical trials, where we're working with contract research organizations to help us work through the FDA. All right, guess we'll keep going.
still not coming.
All right, no worries. So the concept I want to talk about, and this is sort of supposed to be like a quick, tech talk, and so is this idea of smartphone vibrometry. This is a technique that we invented where we can measure the force you're applying to a phone using the smartphone. Now, why does that matter? Well, what's really kind of interesting is there's been a lot of apps out there that says we can measure blood pressure. But all of them, if you ever sort of see an advertisement or read something about it, all they say is put your finger on the camera and we're going to magically tell you blood pressure. And what they do is a technique called pulse wave analysis. They look at the pulse, and the pulse optically is what's called photoplethysmography, it's PPG. And you see this shape. I can draw it out in my sleep, but it kind of looks like this. So there's this pulse in the beginning, and then there's sort of a dip, and then there's a jump next. And so this is created by a reflection of waves that goes through your body. Your heart beats, and then the primary wave goes through your arm, and you'll see that first bump, and then blood also goes elsewhere. It goes under, and it hits the bifurcation in between the legs, and bounces right back up. And so that wave creates this shape. Now, people have sort of postulated that, well, hey, that's a arterial stiffness that has to do with basically you knocking that as hard as possible, and that correlates to blood pressure. Now, that technique only works if you calibrate, it only kind of works if you calibrate it with a cuff. Now, our vision is that we want to be able to send you an app, and you can download that app, and you can measure it without somehow having access to a blood pressure monitor, because if you have access to a blood pressure monitor, who cares then, right? And so our goal is to be able to have an app on the phone and measure your blood pressure without having to calibrate it. And so how do we do that? The technique that we're relying on is this ability to measure pressure you're applying to the finger as you, actually, I don't have enough hands, but when you press down on your camera, the idea is if we can measure the pressure that you're pressing down slowly on the camera, at some point your blood gets cut off. Now, the camera can see when the blood gets cut off by looking at the same thing, the photoplethysmography. But pressure, how do we measure that? And therein lies sort of the technique that we invented at UCSD, where we vibrate the phone using the vibration motor. And then as you press down on it, that pressure dampens that vibration, and we can then measure the pressure you're applying to the phone by simply using a vibration motor and the accelerometer in your phone. And with that technique, we can both measure that pressure that you're applying to your phone. At the same time, we can measure, using a camera, when did it get cut off. And so we've now done the study, at least within our lab and also with UC San Diego Medicine, where we've validated this technique, where we're actually able to see that we can measure if you're hypertensive or not hypertensive. We don't quite do that sort of categorical measurement. We actually do what we call regression, where we can track your blood pressure from both down to 90 mmHg. We've also tested all the way up to 145 mmHg. So our initial study is not all the way to the ends, but it was good enough for us to see that, hey, we can track systolic and diastolic blood pressure. We can track heart rate, but we can't. And now we have been able to bring that out of the lab and got some money from the NIH to actually bring that next step with what I call the CRO, who's going to actually push us through the same ISO standards that all blood pressure monitors actually go through. And so we're working on that. The paper is going to be hopefully published quite soon. So you're actually probably the first people to hear about it. So I'm glad to have shared that with you all with no slides. But I hope you guys were able to use your imaginations. Imagine that phone vibrating in your pocket will one day measure your blood pressure. All right, well, thank you very much.
Sorry about it. I don't know what.
Are you taking investments?
Say again?
Are you taking investments?
Currently, we're working off of the non-dilutive funding, so that's our current goal.
How do you discern between, skin tone and all that? There's, recent published papers of like pulse oximeters not being accurate for, African Americans and those with darker skin complexion.
So the question's about skin tone, and I think that's a super good one, right? The problem for pulse ox and why skin tone really matters is because it's looking at the coloration for different wavelengths. Now, what we're doing is actually looking at the pulse amplitude. And so depending on the skin tone, what you'll do is you'll sort of universally change the sort of the ratio, right? And so because we are measuring sort of when did it get cut off, that ratio of your own skin tone at bigger pulse and lower pulse, we calibrate for that. The main issue is actually whether or not we get enough light to just get that brightness. And so that's the main issue we've been sort of working on. So some phones, it's a really dim screen. So you imagine it's in the front of the screen. So if it's super dim, we have found a little bit of issue. What you can do is turn off the lights in the room, and then it sort of helps. But most modern phone screens are pretty darn bright, probably too bright. And so for the most part, we don't have the issue of getting enough signal. And so that's the main thing we've sort of have dealt with, and we've stared at this quite a bit.
I guess the main question is like, what physical barriers are you still facing? Like, I guess I mentioned skin tone, but what about like skin thickness? Like, I have a lot of patients that have like.
Yeah, I think that's the harder one. That's the harder one for us. And then also, there's some things we've struggled with, which is dexterity. And so usually you don't have to press on the blood pressure monitor yourself and control that. So that's actually probably the hardest one that we've had to sort of figure out. So we had the chancellor here earlier this week, and he was able to do it, but he was like, the UI could be better. Anyone else?
Okay, thanks, Ed. So what we're going to do is we're going to take a 5-minute break here, and then our final panel session for the day, led by Dr. John Madison. And that'll take us to the end of the day. So let's just take a quick coffee break, and while we're getting set up here. Oh my God.